Hello, my name is Jacob Rodriguez, and in Project 2, I will be rhetorically analyzing field artifacts to discuss the clinical implications of CRISPR technology and gene therapy. Before I start analyzing artifacts, I think it is necessary to summarize what CRISPR technology is and how it is used in gene therapy. CRISPR systems are tools to manipulate genomes by cutting certain target sequences in DNA strands and replacing them with the desired sequence. Many researchers have discussed the possible applications of CRISPR gene therapy, as they state that people who are not as genetically gifted may be able to pass down better genetic traits to their offspring after gene therapy, but there are also several ethical considerations regarding these theories. Over the past decade or so, research using CRISPR technology has been conducted to devise possible uses of CRISPR systems to treat and possibly cure certain epidemiological diseases such as sickle cell anemia, HIV, and COVID-19. CRISPR gene therapy consists of three main steps: recognition, cleavage, and repair. The first step involves identifying the target sequence in the DNA strand that is to be replaced. The second step, cleavage, involves cutting the sequence out of the DNA strand, leaving a gap in the DNA. The third step, repair, involves adding the desired sequence to the DNA strand and facilitating the cell's repair mechanism to seal the new sequence into the DNA strand. CRISPR systems have been the main form of gene therapy over the past few years, as it has shown great promise in efficiency and reliability. The first artifact that will be analyzed is an academic review article on CRISPR technology called CRISPR Gene Therapy, Applications, Limitations, and Implications for the Future. I chose this review article because it contains a lot of information on the uses and limitations of CRISPR technology, and it closely relates to my topic of the clinical implications of CRISPR technology. This article does not just focus on one experiment, but reviews numerous studies using CRISPR technology. This academic research article utilizes logos in that it appeals to the logicality of using CRISPR technology in clinical practices, with research to support the author's claims about the possible uses of CRISPR gene therapy in the future. This article discusses not only the mechanisms of CRISPR technology, but also its current and possible future clinical practices and implications of these practices. This article also appeals to the logicality of using CRISPR technology by addressing possible limitations and ethical considerations of CRISPR technology. The first quote displayed towards the bottom of the page is an example of this, as the writer claims that there are many possibilities for the use of CRISPR gene therapy, but there are also several technical and ethical considerations that need to be addressed for clinical practices. This article, as well as many others, comments on the reliability and affordability of CRISPR technology compared to past technology used for gene editing, and states that the CRISPR system has surpassed the advancements of previous technology due to its relatively low price, ease of access, and efficient and precise performance. Pathos is utilized in this article to portray a sort of hopeful yet neutral mood, as the writer uses positive language in support of CRISPR technology, but also addresses the limitations of this form of gene therapy. The first quote that was discussed earlier, that states the recent advent of CRISPR technology in clinical trials has paved the way for the new era of CRISPR gene therapy to emerge. However, there are several technical and ethical considerations that need addressing when considering its use for patient care. This quote portrays a positive mood for CRISPR technology, but then switches its language to put a check on this mood and addresses potential concerns about the use of CRISPR technology in clinical trials. The writer uses this structure in numerous sentences in different parts of the article to put CRISPR CRISPR technology in a positive light, but also note that there are ethical considerations among other possible concerns that must be addressed. Ethos is employed in this article as the authors of this review article are credible and have proven to have extensive knowledge and research on the topic of gene manipulation and the use of CRISPR technology. One of the authors, Fatima Uddin, is a student of the Downstate Health Sciences University pursuing a doctorate degree. Charles M. Rudin is another author of this article, who is a doctor/researcher at the Memorial Sloan Kettering Cancer Center in New York and received his doctorate degree at Cornell University. The third author is Triparna Sen, who has received a doctorate degree from Cornell University as well, and is an assistant professor at the Icahn School of Medicine at Mount Sinai in New York City. This article is also credible because it is a review of previous research using CRISPR technology to edit genomes and has itself been reviewed by numerous researchers. An artifact is a non-text-based artifact that depicts the general idea of gene manipulation. I chose this image as my second artifact because it depicts the general idea of gene manipulation and contains a lot of content that can be unpacked. The image demonstrates the functionality of DNA and overall health and general functions such as heartbeat, brain functions, etc. The diagrams adjacent to the DNA strand that is being manipulated portray the fact that DNA contains all of the information about our bodies and how they function. The inclusion of hands in the image actively cutting and moving parts of the DNA show that, in general, gene manipulation is a very hands-on procedure, even with the use of CRISPR technology, as there are multiple steps involved in the gene editing process. In the image, it can be observed that a hand is using surgical scissors to cut part of the DNA strand. This is a simple demonstration of how the DNA strand is cut, but with CRISPR technology. The CRISPR machine cuts the DNA strand into double-stranded breaks since it recognizes the target sequence through its complementary base pair component. The orange color of one part of the DNA strand shows that it is most likely a new or foreign sequence of DNA that has been essentially manufactured and is being placed into the parent DNA strand to heal the gap in the strand. During the CRISPR gene editing process, this step is considered to be the repair step, when the non-homogeneous and joining is used at the site of the gene editing to facilitate a cellular repair mechanism. Overall, CRISPR has changed the way people understand gene therapy, and genetic manipulation has entered a new era. CRISPR shows immense promise in its applications to clinical practices, but before it can become completely mainstream, there are numerous possible limitations and ethical considerations that must be addressed. These artifacts have been very useful in allowing me to understand CRISPR systems and their possible applications in clinical trials.